Hi, my dear students. Happy morning to all. Myself, Anu Shashidharan, Academic Director and Biology Faculty of Rankings Entrance Training Academy. Today, I am going to talk about an important process, that is fertilization. As you know, fertilization is the most important event in all type of sexually reproducing organisms. Double fertilization is the unique event in flowering plants or angiosperms. Today we are going to discuss in detail the various steps in double fertilization of angiosperms. As you know, just after pollination, there is a continuous dialogue between the pollen grains and the stigma to identify whether it is a compatible or incompatible pollen. The process we have discussed in the last video, what we can say, pollen-pistil interaction. After pollen-pistil interaction, what happened? The pollen grains germinated to form a tube-like structure called germ tube or pollen tube. The pollen tube actually moves through the style and finally enter into the ovule or embryo sac. As you know, inside the embryo sac there is an egg apparatus which has two synergids and one egg cell. The synergid actually having a cellular thickenings that we have discussed. It is called a filiform apparatus. Filiform apparatus plays an important role to guide the pollen tube directly into the embryo sac, especially into the synergids. After the entry of pollen tube, what happens? The pollen tube releases two male gametes into the cytoplasm of the synergids. Okay, the pollen tube releases two male gametes into the cytoplasm of the synergids. Now the situations are ready for fertilization. One of the male gametes moves towards the egg cell and fuses with its nucleus to form a structure called a zygote. Is it clear? Means one male gamete fuse with the egg cell to form a diploid structure called a zygote. This is the zygote. The process is called a syngamy. The other male gametes moves towards the secondary nucleus and fuse with it to form a triploid structure called a primary endosperm nucleus. The process is called a triple fusion. So, in angiosperms, during fertilization, two fusions are there, syngamy and triple fusion. Hence, the process we can call it as double fertilization. So, what is the product of double fertilization? A diploid zygote and a triploid primary endosperm nucleus. After fertilization, the zygote later develop into embryo and the primary endosperm nucleus later develop into endosperm. This is something about double fertilization process occur in angiosperms. Next phenomenon we are going to discuss is called outbreeding devices. So what is the meaning of outbreeding devices? Suppose you think about if a plant performing continuous inbreeding. What is the meaning of inbreeding? Inbreeding means self-pollination. If a plant performing continuous inbreeding, what happens? The plant leads to an important stage. The stage is called inbreeding depression. So what is the meaning of inbreeding depression? Inbreeding depression means the continuous inbreeding usually reduces the fertility and even productivity. This condition is called inbreeding depression. Some plants they are having some devices to encourage cross pollination and discourage self pollination. Okay, this is actually some adaptive features present in certain plants to help the process of cross pollination. Such devices we can collectively call the outbreeding devices. First one, that is the pollen release and stigma receptivity are not synchronized. So what's the meaning? The pollen release and stigma receptivity are not occur at the same time. Means either the pollen is released before the stigma becomes receptive, or the stigma becomes receptive much before the release of pollen. Is it clear? Second one, the anther and stigma placed at different positions. Is it clear? So that the pollen cannot come in contact with the stigma of the same flower. Third one is that self-incompatibility. What is self-incompatibility? This is actually a genetic mechanism and prevents self-pollen from fertilizing the ovule and inhibiting pollen germination or pollen tube growth in the pistil. Okay, this is called a self-incompatibility. Another device to prevent self-pollination is the production of unisexual flowers. The condition is called diclining. If both male and female flowers are present on the same plant, such condition is called monoecious, you have studied. It prevents autogamy but not geitonogamy. This condition commonly seen in plants like castor and maize. In several species such as papaya, the male and female flowers are present on the different plants. That is, each plant is either male or female. This condition is called dioecious condition, which prevents both autogamy and geitonogamy. This is something about outbreeding devices. One more concept we are going to discuss now, that is artificial hybridization. Artificial hybridization is defined as the process of crossing two genetically different individuals having desirable traits to obtain an offspring having superior traits than the parents. This is one of the major approaches of crop improvement program. Two important steps in artificial hybridization, that is emasculation and bagging. In artificial hybridization, as you know, the first step is the selection of parents, that is one male parent and one female parent. After the selection of parents, what we have to do? We have to collect the pollen grains from the male plant that should be transferred to the stigma of the female flower. The process is called artificial pollination. Is it clear? If the female parent having bisexual flower, we have to remove all the stamens by using a scissor. The process is called emasculation. Emasculation helps to prevent additional pollination or further pollination by any other insects or pests or self-pollination. After emasculation, we have to cover the female flower by using a polythene bag. This is actually doing to prevent the further pollination by wind and insects. Afterwards, what happened? All the post-pollination events must be occur. That means the fertilization process, formation of embryo, formation of endosperm, etc. Finally, we can collect the seeds. The seeds on germination, we will get new plants, which is commonly called hybrids. The hybrids may contain or may have both the characteristic features of male parents and female parents. And one more important point, you are very familiar, the process of emasculation. I said the process of emasculation means the removal of anther from a bisexual flower by using a scissor. Emasculation really prevents the self-pollination, means the further pollination. If the female parent produces unisexual flowers, there is no need for emasculation. Okay. So I am going to wind up here. We will meet in the next video with other topics, that is the embryo development and endosperm development. Thank you.